Julia Beck. Julia is a sixth-year PhD student in the biochemistry department in the College of Science. Her faculty advisor is Professor Francis Castellino. And Julia today will be presenting on A Tale of Two Proteins.
It was the best of times, it was the worst of times. Today, I'm going to tell you the tale of two proteins. The first protein is called plasminogen, and it is floating through our bloodstream right now. And it is involved in wound healing. When you get a cut, a clot is formed in order to prevent a lot of blood loss. But eventually, once this wound is healed, the clot needs to be removed. Plasminogen helps heal the body by first becoming activated, and then it can remove the clot like a little Pac-Man. This is the best case scenario for us, but things can go quickly bad when another protein gets involved. This other protein is called PAM, and it is found on the surface of the bacteria Streptococcus pyogenes, a human-specific bacteria. This bacteria commonly causes strep throat, but it can also cause necrotizing fasciitis, also known as the flesh-eating disease. A few years ago, a man was walking along the coast in Florida, and he accidentally cut his toe. The next day, he was in the hospital and had part of his leg amputated. Part of what makes this bacteria so dangerous is the interaction of these two proteins, the bacterial protein PAM and the human protein plasminogen. My thesis is to better understand how these two proteins interact and why when they do interact, the bacteria become so dangerous and in some cases deadly. I have isolated these two proteins, and in doing so, I'm able to study the dynamics of this interaction, measure the activity of plasminogen, and actually see how these two proteins interact. Our lab has found that PAM interacts with plasminogen in a new way, one of which we haven't seen before. My work has found a potential mechanism for this interaction. This non-traditional interaction is fascinating because it demonstrates how the bacteria are able to both adapt and evolve based on their surroundings. While it is important to understand how these two proteins interact, it is equally important, if not more so, to understand why this interaction causes the bacteria to become so dangerous. I have discovered that when PAM interacts with plasminogen, plasminogen is more easily activated. Once plasminogen is activated, it can then break down the barriers that normally prevent the bacteria from invading throughout the body. Thus, when PAM interacts with plasminogen, the bacteria are able to spread throughout the body and eat our flesh. So by understanding the specifics of this interaction, we can eventually develop drugs blocking this interaction, weaken the bacteria, and allow us to continue to enjoy the best of times. Thank you.
Thank you, Julia.